Session remains on time. You can submit your questions via the app or the online conference platform, and you can also ask questions in person here by queuing behind the mic. When you are submitting questions online, please make sure to mention your name if you don't want to remain anonymous, and also please indicate who is your question for. Thank you.
Okay, then I think we can start right away, and it's my pleasure to introduce our first speaker. It's my co-chair, Sarah Mirza. She already said it, she's from the Sunnybrook Research Institute in Canada, and she will present on association of low-frequency variants with regional cortical gray matter volumes in genetic frontotemporal dementia, results from GenFi.
Thank you, Michelle. Thank you, AAIC, for giving this opportunity. I have nothing to disclose. As we all know, frontotemporal dementia is a group of rare neurodegenerative disorders which are heterogeneous clinically, genetically, and pathologically. The age of onset is 45 to 65 years, so it's affecting people who still might be working on having familial or financial responsibilities, and that is why FTD is a huge societal burden. As the name implies, the disease primarily affects frontal and temporal lobes of the brain, and people can present with behavioral impairment or language impairment with or without disturbances of the motor function. About 50 to 80% of FTD is sporadic, but 20 to 50% is familial, and it is due to autosomal dominant mutations in any of the three genes, MAPT, progranulin, and C9 open reading frame. There are two main pathologies which are observed on autopsy. One is a tau positive associated with MAPT, and the other is tau negative, or the TDP43 pathology, which is associated with progranulin and the C9orf open reading frame repeat expansions. There is substantial clinical heterogeneity in genetic FTD. Heterogeneity within the families is also very common with multiple presentations. And clinical heterogeneity is also present within the same family, and that is in part due to the variable involvement of the hemispheres or the lobar regions. However, it is not known if one hemisphere or one lobar region is affected more than the other. Finally, the age of symptom onset is highly variable, particularly those who have granulin mutations or C9orf open reading frame repeat expansions. That is why we need to identify modifiers of genetic FTD that alter the natural course of the disease, and rare genetic variants with larger effects could be such modifiers accounting for the missing heritability. That is why in this study we aim to identify rare genetic variants that associate with regional cortical and subcortical gray matter volumes in genetic FTD. We hypothesize that rare variants other than the autosomal dominant mutations contribute to imaging endophenotypes, and this contribution could be through independent functioning of these variants, interaction of these variants with the causative FTT mutations, or interactions of these variants with each other, or a combination of these. The study is embedded within the GenFi consortium, which is a multi-center consortium for tracking the evolution of genetic FTD. Currently, 40 sites are part of GenFi consortium from 15 countries across Europe and Canada. GenFi recruits cases and unaffected members from families segregating genetic FTD. I show this diagram here to demonstrate that the current study is part of a mega genomics project embedded within GenFi, which involves both common and rare variants. However, today I would just focus on the rare variants in relation to regional cortical and subcortical gray matter volumes. I also presented a poster on Tuesday relating to the cognitive outcomes, if anyone of you had a chance to look at it. This is a busy slide, but I would just emphasize on the numbers. We started with 593 genotyped individuals, and after a very strict quality control, we ended up with 565 individuals, but the final sample is 517 because that was the imaging phenotype and genotype overlap. For the variant level, we started with around 459,000 genotype variants which were typed on the neurochip. After a strict quality control and imputation on top med, we filtered on rare variants, that is a minor allele frequency of 1%. And then we applied a loss of function annotation, which left us with 67,000 variants to be tested. We tested population stratification, which is very important in genomic studies, by multidimensional scaling, and we calculated principal components to be included as covariates in the linear regression analysis. However, I would like to mention that the population was largely homogeneous Caucasian, with only 3 outliers who were removed before any analysis. So we tested a large number of regions, and here I show the list of regions tested just to give you an idea. The volumetrics were collected by T1-weighted scans performed on 3T and 1.5T scanners. It was a linear regression analysis, and the covariates included our age, sex, kinship matrix for the relatedness, total intracranial volume, mutation carrier status, principal components to account for the population stratification, and the scanner site. We defined rare variant as minor allele frequency less than or equal to 1%, and the significant threshold was 1.8 to 10 raised to the power minus 6, accounting for the number of regions tested and the number of genes tested. We carried out burden tests, which actually test aggregate burden of rare variants within a gene in relation to a phenotype of interest, and we used annotation for loss of function variation. This table shows the baseline characteristics of the sample, but I would only like to highlight here that this sample included around 40% of non-carrier controls, 43% pre-symptomatic carriers, and around 17% symptomatic carriers of genetic FTD mutations. This is again a busy table, but it is more to highlight the regions which popped up as significant and not the names of the genes, because they don't mean anything with these abbreviations. If you see the frontal regions, temporal, some parietal regions, and a few subcortical areas had significant hits. In total, we had 58 genes in 23 regions, and there were overlapping genes. Here I show the genetic hits for the frontal, temporal, and parietal lobes. Collectively, the molecular function of these genes is binding and transcription activity, and they belong to the protein classes of defense protein, transcriptional regulation, and again, binding. For the regions which had multiple hits, I take the example of the left temporal lobe volume, which had several genes which were significant. Collectively, if you see, their molecular function is also for binding, regulation, transcription, or transporter activity. We also did some interaction analysis. We showed that these genes actually have physical interactions with each other, and some of these also co-express. So the identified genes may function independently or via co-expression, shared protein domains, or interactive or synergistic pathways in addition to interaction with the causative FTD mutations. So we are doing several post-hoc analyses which are focusing on identifying the most significant SNPs within these genes identified, testing interactions of these identified SNPs with autosomal dominant FTD mutations, and candidate gene analysis with the strongest candidate SNPs. Finally, the further steps are replication of these results followed by functional studies. In conclusion, identification of rare variants contributing to disease phenotypes may help identify genetic modifiers of familial FTD, and knowledge of such modifiers of genetic FTD that alter the natural course of the disease can help design better trials and with better informed outcome measures. I would like to thank my supervisors, my co-authors, the GenV Consortium, GenV participants, and our funders. Thank you very much.
Okay, so we jump straight to our next speaker, who's David Whiteside from the University of Cambridge, and the title of his talk will be Functional Connectivity Moderates the Impact of Synaptic Loss on Behavior in Frontotemporal Lobar Degeneration Syndromes. Thanks.
Hello, I'm David Whiteside, and I want to thank the organisers for the opportunity to talk about our work, which has looked at the connection between synaptic PET and functional connectivity in syndromes associated with FTLD. So no disclosures. So as we know, the macroscopic neuropathological finding of frontotemporal lobar degeneration is associated with a heterogeneous range of clinical entities and also varied underlying clinical proteinopathy. But they all have in common synaptic loss. Preclinical models of neurodegenerative disease have suggested that synaptic loss occurs early, and postmortem study in FTD suggests that the synaptic loss is extensive and correlates well with clinical severity. For a long time, we've had an indirect measure of synaptic health with FTG PET, but more recently, a number of groups have studied using UCBJ, which binds to the synaptic vesicle glycoprotein 2A in a number of neurodegenerative conditions. I'm just going to briefly overview some of the work we've already published looking at these conditions before talking about the connection to connectivity. So particularly focusing on BVFTD and the 4 repeat tauopathies, amyloid negative corticobasal syndrome and PSP Richardson syndrome. So this work led by my colleague Megan Holland found extensive synaptic loss in PSP and amyloid negative CBS, and that the amount of synapse loss correlates well with clinical severity. In BVFTD, led by Mauro Malpetti, we found extensive synaptic loss, particularly in the frontal lobe in behavioral variant FTD. And again, this correlates well with severity. So I wanted to ask, given these findings, how synapse loss may affect networks in these conditions. So to characterize networks, we use resting state functional MRI. For synaptic loss, our focus was really on UCBJ, but we also looked at a complementary diffusion-based modality called NODI. We characterized atrophy with volumetric MRI, and our key outcomes were the Addenbrook's cognitive examination, which is a marker of general cognitive health, and a PSP rating scale. So this is our key question. Does synaptic loss affect cognition, and is there an interplay with connectivity? Our aim is really to better characterize the factors that determine presentation and progression, potentially supporting translational medicine experimental studies. So our cohort was 79 participants, 24 controls, 29 individuals with PSP Richardson syndrome, 16 amyloid negative cortical basal syndrome, and 10 people with behavioral variant FTD. There were no differences in age or sex between groups. And as I described as multimodal imaging study with dynamic acquisition for the UCBJ PET. So I characterize connectivity in two distinct ways. Firstly, using weighted degree, which is the sum of connections to each node. This is with the Hammersmith parcellation. There are some differences in parcel size for that atlas. So for the degree, we subparcel to be approximately equal-sized parcels. Just on the left, as you look at it, I've just put mean weighted degree for patients, Z-scored relative controls compared to regional binding potential of UCBJ, again in patients. And there's a relationship with p-values counted, with permutation testing, with spatial autocorrelation being preserved. We also looked in mixed linear effects models with a random intercept for participant for region and a slope within UCBJ for region, and found a relationship between weighted degree and with UCBJ in all patients in each of the clinical syndromes considered individually to a greater degree in patients than controls and beyond age, sex, and fMRI motion, and also beyond atrophy. Just looking separately region by region in linear model with covariates, we compared the different modalities to see, and everything showed here is FDR corrected p less than 0.05. So if the two on the left, the markers of synaptic health, UCBJ, and orientation dispersion index from NODI, predicted more areas of connectivity than grey matter volume. If you combine all three modalities using non-parametric combination testing with adjustment for the extra contrast, you get a better improvement, better prediction. So having established that there's a relationship between synaptic density and with connectivity, we wanted to capture a small number of components to describe variation, regional variation in synaptic density to take on to compare with cognition. To do this, we use an independent component analysis of the concatenated UCBJ mats. Of 10 components, we discarded 3, and 6 of the remaining 7 differed by group. These showed variation not just from controls, but also within phenotypes. In general, synaptic loss was greater in the behavioral variant FTDs, but this was more noticeable in some components. So if you look at component 4 with focus on frontal lobe and the insular cortex, that had profound reduced synaptic density in BVFTD, whereas component 2 with a more posterior distribution had greater loss in CBS and PSP. We then took those components as templates and asked what the connectivity of those regions was. We then could compare what each per participant, how they loaded onto the UCBJ component with their connectivity. So we can ask that voxel-wise. So you see the orange areas are all there's a significant association with their connectivity and with their synaptic density per participant. This is true in all participants, in patients alone, and beyond important covariates. So therefore, we have a score per participant for their synaptic density for these regions, and also a score for how much they connect to those regions. So we wanted to then go ask, how does the interplay to affect cognition? We use model selection with a baseline model putting the interactions between the UCBJ components and their related fMRI components, and then using Bayesian information criteria, to ask what the best model was. And that included a combination of UCBJ and fMRI components and interactions between the two, with stronger relationships seen between synaptic density and connectivity, where there is relatively maintained cognition. We found a similar finding in using the PSPRS, with best prediction being a combination of striatal loss of synapses and moderated cortical loss of synapses. Given that we found connectivity changes both at the site of the components, so both at the site of the synaptic loss and distally, we also wanted to ask, was there a difference in how connectivity mediates? We found with certain components that the connectivity within the region, there was evidence of mediation, particularly in the behavioral variant FTDs, where the connectivity drives, where there's connectivity mediates the result of the synaptic loss. So key takeaways are that the synaptic loss in FTLD syndromes did show regional specificity by syndrome. It correlated well with functional connectivity and beyond atrophy. It affects clinical severity via both co-localized and remote connectivity loss, and shown a way that we can combine different modalities to model how various factors may determine cognition. The various people who have been involved in this study, particularly the volunteers and families and the leaders of the project, Professor Rowe and Professor O'Brien, and also my supervisor, Dr. Rittman. Thank you.
Thank you very much, David. Our next speaker for today is Michel Groth from the Institute of Biomedicine, Seville, Spain, and he's going to talk to us about clinical and biomarker characteristics of amnestic MCI patients exhibiting a temporal limbic FDG PET pattern suggestive of late. Michel, stage is yours.
Thanks. Nothing to disclose. Limbic predominant age-related TDP-43 encephalopathy is a frequent neuropathologic finding in advanced age that associates with simple complex sclerosis and associates also with amnestic deficits that may mimic AD dementia clinically. So this is shown here in this in autopsy findings where a considerable subset of patients clinically diagnosed with AD dementia actually turn out to have TDP-43 pathology as their principal pathologic correlate. And this condition, late, is associated with advanced age, comparatively slower global disease course compared to AD, and a somewhat more amnestic selective cognitive deficit. It has also been linked to some specific genetic risk factors, such as the TMEM106B risk allele that had originally been identified in FTLD TDP-43. And it's associated with a lower frequency of the ApoE4 allele, at least when compared to AD dementia patients. In our previous study, we wanted to assess the utility of FDG PET to detect this condition in vivo, since we are really lacking today, still lacking validated biomarker that would allow us to be used for differential diagnosis between AD and late during lifetime. And to assess the utility of FDG PET, we first used antemortem FDG PET from autopsy-confirmed cases to demonstrate that late patients are actually characterized by a very specific temporal limbic signature of cortical hypometabolism that's markedly different from this typical AD typical tempoparietal pattern. And we then used these patterns in combination with a pattern matching approach based on spatial correlation to screen a larger cohort of almost 250 patients clinically diagnosed with AD dementia to search for those patients who may have an individual FDG PET pattern that may be more suggestive of late than of AD. And this was the case for approximately 10% of the cohort. And when we compared these patients to those with a more AD typical hypometabolic pattern, we found that these late-like patients showed several features that were consistent with an underlying late neuropathology. So they were considerably older, on average 10 years. They had a more amnestic predominant cognitive profile as indicated by this cognitive profile variable, which contrasts memory scores to executive function scores. And they had similar degrees of cognitive impairment at baseline, but showed a considerably slower disease course over follow-up. So all features consistent with late. They also, underlining the idea that it may be actually late pathology driving the clinical syndrome in these cases. They had also significantly less AD biomarkers, less abnormal AD biomarkers, and they were also less likely to be ApoE4 carriers, so 50% ApoE4 positive versus almost 70% in the AD-like cases. But on the other hand, they were enriched for the TMEM106B risk allele, which is associated with TDP-43 pathology. So in the current work, we wanted to follow up on this initial study to assess these late versus AD-like FDG patterns for patient stratification at an earlier pre-dementia disease stage. And we also wanted to have a closer look to the association between this late-like FDG pattern with regional tau pathology. So for that, we studied all amnestic MCI patients currently enrolled in the ATNI study who had a baseline FDG PET scan available, about 900 patients. And we then classified the individual FDG PET scans using this pattern matching approach, which matches individual scans to these autopsy-confirmed late and AD-specific hypometabolic signatures. And then the classified patients group were then compared with respect to longitudinal cognitive outcomes, ApoE4 and TMEM106B SNPs, and CSF biomarkers, and also tau PET using fluortausobir, which was available in a subset of the sample. Okay, so first, when doing the classification of the individual scans, we first found that a large portion of these amnestic MCI patients did not have sufficient degrees of regional hypometabolism that could be used for this pattern matching approach. And then others had a distribution of hypometabolism that was not consistent with either the AD specific or the late specific signatures. But then finally we ended up with 144 patients being classified as having a typical atypical temporal parietal hypometabolic pattern, and this is their average image. So it seems this automated classification approach does a pretty reasonable job. And 105 patients who showed this very distinct temporal limbic pattern, most pronounced here in the medial temporal lobe and surrounding limbic structures. And when we then again look at some of the clinical features of these patient subgroups, we could reproduce here that these amnestic MCI patients with a late-like FDG PET pattern were considerably older. And so on baseline, these groups were basically indistinguishable, but the late-like patients showed a considerably slower disease course over follow-up. Again, they were also had significantly less abnormal AD biomarkers and cerebrospinal fluid, and this was particularly pronounced for tau pathology biomarkers. Consistent with our previous study in AD dementia or amnestic dementia patients, we again found that these were significantly less likely to be ApoE4 carriers, but they were again these late-like patients were again enriched for the TMEM106B risk allele. We then wanted to have a closer look on regional tau pathology in these subgroups, and here we found that these late-like patients had lower levels of tau PET uptake across all examined ROIs, and this was most pronounced in these extramedial temporal ROIs, so temporal and parietal neocortical areas, where we found that elevated tau PET signal in these areas was actually very rare in these late-like patients. And this is interesting because these are supposed to be like tau deposition in these areas is supposed to be more closely linked to clinical symptoms. We finally also did some individual level analyses using visual reads performed by my colleague Alexis Moscoso, who I don't know if he's here today still with us. And so this is a tremendous work, like visually reading thousands of tau PET scans. And using this information, we found that amnestic MCI patients who had this clear temporal parietal pattern of hypometabolism suggestive of AD, actually almost all of them were had a positive tau PET read, whereas the majority of these late-like patients had a negative tau PET read. Also, there was a subset, like 40% that at least have moderate levels of tau pathology in this visual read. And so we think that this could reflect a comorbid condition or maybe limbic predominant hypometabolic subtype of AD. Okay, so in conclusion, we could show that amnestic MCI patients with temporal limbic FDG PET pattern suggestive of late show several clinical and biomarker features that clearly deviate from prodromal AD. And this includes a considerably older age, a slower disease course, less abnormal AD biomarkers. They are less likely to be ApoE4 carriers, but they are enriched for the TDP43-associated TMEM106B SNP. A subset of these patients do show advanced tau PET signal, and well, these could overlap with a limbic predominant FDG PET subtype of AD that has previously been described. So as an outlook, I think it would be nice to have more autopsy data to get to a better characterization of these how early late versus AD typical FDG PET patterns actually look like, and using such early versions maybe of these patterns could increase the sensitivity for early detection of these conditions at the pre-dementia disease stage. Further work needs to be done for distinguishing these patterns from other medial temporal focused pathology, limbic predominant AD part and AGD. With that, I'd like to thank all my colleagues at the IBIS in Sevilla, especially Jesus Silva, who's a postdoc in my group and did most of these analyses, actually, my international colleagues from the University of Gothenburg, funding sources, and all of you for your attention.
Thank you very much, Michel. Our next speaker for today is Brian Gold from the University of Kentucky, USA, and he's going to talk to us about plasma TDP43 levels are associated with neuroimaging measures of temporal lobe structure.
Okay, thank you so much to everybody who's still here and is getting through this final event together. And thank you for the last presentation, which really helped me set up the concept of late. So I have no disclosures, and as was stated, late is a unique form of dementia that is caused by TDP43 pathology. It's often referred to as an AD mimic because it has a predilection for medial temporal lobe regions, and it also causes episodic memory decline in early phases of the disease. So again, often referred to as an AD mimic, but what we can see here, this is a recent paper from the University of Kentucky at our ADRC from Ruth Nelson. And if you look at the Venn diagram, what you see is that, yes, indeed, late typically overlaps at pathology with AD. But you can also see at the bottom there that there is, let's see if I can point here. So there is a unique group that has a single proteinopathy of late, and that can present itself. And so we need to understand more about late and to develop biomarkers to look at it. And one of the reasons why is that when the studies have shown is that basically is that TDP43 pathology goes up, cognition goes down, and that's independently or after sort of controlling for other pathologies. So late, it can present as both a pure, unique form and is very important for cognitive function. So basically, that sets up the idea that we really need in vivo biomarkers for late. And previous presentation was fantastic, and it looked at FDG PET. However, there's no specific ligand right now for late, for TDP-43 pathology that is commercially available and validated. However, we have plasma available, and plasma TDP-43 has actually been associated with AD. It's higher in AD than in controls, and it's been correlated with function or level of pathology in FTD, although not with Alzheimer's disease. However, as a first step, we really need to assess plasma and potential markers given that they're non-invasive. So, so basically, we're starting here with this premise, with the idea that essentially, if the plasma TDP-43, and we're looking at total plasma TDP-43 here, is valid, it should be correlated with neurodegeneration in regions of the brain that we know are associated with early signs of late, namely limbic regions. Okay, so the goals here were really to identify the neurodegenerative correlates using MRI of TDP-43 plasma. We had a two-prong approach to this. We had an original ROI-based hypothesis-driven analysis. And this is based on looking at 3 regions in the medial temporal lobes that have been associated with early stages of late on pathology, and that is the amygdala, the entorhinal cortex, and the subiculum of the hippocampus. So in addition, then, we did exploratory analyses across the entire cortex using cortical thickness. All right, so our subjects come from the University of Kentucky ADRC longitudinal cohort, and for this particular study, we selected individuals who had banked plasma and an MRI T1 weighted sequence within 1 year of each other. And so plasma was collected in the typical manner, and scanning was performed on a 3T Prisma scanner at our center at the University of Kentucky. And the main sequence of interest here is the T1 weighted MP range. Okay, so for our analyses, all the analyses were done at the UK Biomarker Core, which includes both a fluid and imaging component. The fluid analyses were done in Donna Wilcox's lab, and the imaging analyses were done in my lab. And for the imaging analyses, for our hypothesis-driven analyses, FreeSurfer was used for segmentation, and then we got volumes of specific structures and looked at their correlation in SPSS, looked for regression models to see if plasma TDP-43 predicts the volume in these different regions. And then, as I mentioned, for the exploratory analysis, we're looking across the vertex with cortical thickness. Okay, so here is the set of subjects that had plasma and MRI within 1 year of each other, and there are 72 of them. Now, I will note that a subset of these participants had MCI, and I know a lot of people will say, oh, how about if you look at the MCI separately? Is that different than the normal control group? And it may be, but unfortunately at this time we really don't have the ability to look at that because we only have a small sample of MCI subjects. So that's something we are going to definitely look at later. And the paper was recently accepted if you want to have a look at it. So I want to mention that all the analyses I'm going to be presenting are done by an outstanding scientist in my lab named Chris Bauer, and he's done these analyses that I'm going to present here, and it's great to have him in the lab. So he ran these regression analyses, and what he found was basically that one of these regions, the ERC, showed a relationship with plasma TDP-43 after Bonferroni corrected comparison, multiple comparisons correction for the 3 ROIs that we had. And when we look at the scatter plot, we can see that it is indeed a negative relationship. As plasma TDP-43 goes up, ERC volume goes down. And I'll also mention that after controlling for plasma markers of AD, such as A-beta 42/40, the relationship persists. So this is showing a relationship that is associated with plasma TDP-43 and cannot be accounted for by AD biomarkers. Okay, so what I'm showing you here is actually the same scatter plot I just showed you. So if we look at that here, what we see again is we have TDP-43 plasma on the x-axis and entorhinal cortex volume on the y-axis. And what I'm highlighting here is in this corner here, what we have is individuals who have high plasma TDP-43 and low entorhinal cortex volume. So all we did here is one thing. We split them using a median split for their AB 42/40 ratio. And keeping in mind that a high ratio is good and a low ratio is bad, we have these subgroups now. So we have individuals who are in red. These are individuals who are at high risk for AD. But the individuals in blue have the same pattern of high plasma TDP-43, low entorhinal cortex volume that cannot be accounted for by having high AD risk. So these are individuals in blue are at high risk for TDP-43 late, and we see some of their characteristics here. And this is just to really say that this type of approach may be useful to us in terms of trying to develop biomarkers to stratify for clinical trials so that we can get people with TDP-43 related dementia in vivo into the right clinical trial, as well as individuals with AD, excluding TDP-43. Okay, so as for the cortical thickness analyses, what we found was that there were 3 regions that showed a relationship with plasma TDP-43. As plasma TDP-43 goes up, cortex thickness goes down, thinner cortex. However, after controlling for plasma markers of AD, what we see is shared variance soaks all that up, and what we have is a single region in the posterior portion of the parahippocampal gyrus that shows a unique relationship with TDP-43. And I was very interested to see in the previous presentation that the FDG signal was also primarily in limbic regions, including, it looks like, this region. And so in terms of a next step, this is just showing the same finding on a less inflated brain. And so, we'll be collaborating with neuropathology. Our neuropathology core, Pete Nelson, runs this, and he's one of the co-discoverers of late. And so, I asked him, is there any pathology here in this region? He said, we don't know, we don't look there. So he's going to look there and see if there is any TDP-43 pathology that would correlate with the imaging findings that we have, which I think is really cool, if imaging can occasionally inform pathology instead of always just us doing what they find. So this is one last thing I'll show. We also want to try to dissociate or take out people at risk for TDP-43 from people who have more pure AD. And so this was an analysis that looked at the entire default mode network and finds a correlation with the plasma markers of AD, but not TDP-43. So this is with a larger set of subjects, and it's not ready yet, but it's something we're going to keep looking at in the future. And in conclusion, the main conclusion is plasma TDP-43 essentially is correlated with neurodegeneration in regions of the brain that we would expect it to be in based on the path record. And basically, plasma TDP-43, these results warrant further investigation of plasma TDP-43 in our analyses, considering late. And I'm being blinked here that I'm 4 seconds over, so I will just thank members of my lab. In particular, I've mentioned Chris Bauer, but Valentino Zachariou also did a lot of free surfer analyses. I want to thank people at our Alzheimer's Center, particularly Pete Nelson and Greg Jaika, who got me interested in this area, people at our MRI Center, as well as the funding, and in particular the scan supplement to our ADRC P30, which is what funded this research I'm showing you today. Thank you for your attention.
Okay, is this working? Yeah. So let me introduce our next speaker then, who's Farwa Fahim from the Boston University Jovanian and Avedisian School of Medicine. And title of her talk is Cognitive, Functional, and Neuropsychiatric Correlates of Regional Tau and Autopsy-Confirmed CTE.
Thank you. My name is Farwa, and I'm from the Boston University CTE Center. I don't have any disclosures. Chronic traumatic encephalopathy is a neurodegenerative disease caused in part by exposure to repetitive head impacts from contact and collision sports, physical violence, and other sources. At this time, CTE can only be diagnosed at autopsy using published neuropathological criteria. And a neuropathological diagnosis of CTE requires perivascular hyperphosphorylated lesions in neurons with preference for the cerebral sulci. In stage 1, isolated p-tau fibrillary tangles are found in the frontal cortex. In stage 2, they spread to the adjacent temporal cortices. In stage 3, they're distributed in the medial temporal lobe structures. And in stage 4, we find diffuse p-tau lesions and neurofibrillary tangles distributed throughout the cerebral cortex, with pronounced neurofibrillary degeneration in the medial temporal lobes. CTE can only be diagnosed at autopsy, but there is a clinical syndrome known as traumatic encephalopathy syndrome, or TES, and the TES criteria are used to determine the level of certainty of underlying CTE pathology. A diagnosis of TES requires substantial exposure to repetitive head impacts, the presence of core clinical features that include cognitive impairment and a neurobehavioral dysregulation. There must also be a progressive course in which these symptoms get worse over time. There are some supportive clinical features as well that include mood and psychiatric symptoms like depression and apathy. And these clinical features should not be fully accounted for by any other neurologic, psychiatric, or medical conditions. The current criteria were developed without biomarkers, and due to the heterogeneous nature of these symptoms, it's currently unclear if they are related to CTE. We know that p-tau, or hyperphosphorylated tau, is a precipitant of neurodegeneration and the resulting cognitive and neuropsychiatric decline in Alzheimer's disease and related dementias. Here we wanted to see if there was an association between the clinical symptoms seen in TES and the p-tau pathology found in CTE. We did that by correlating informant-based scores on standardized scales of cognitive, behavior, and mood symptoms with postmortem ratings of p-tau by neuropathologists, and expected to see that higher scores suggestive of probable impairment were associated with greater p-tau pathology severity. The brain tissue we used came from 364 brain donors that were diagnosed with CTE at autopsy and were a part of the Understanding Neurological Injury and Traumatic Encephalopathy, or UNITE brain bank, which is a brain bank dedicated to people who've been exposed to repetitive head impacts. Once we receive the tissue, it is hemisected, and part of it is fixed and divided into blocks that represent the regions important in CTE. The tissue is then stained and examined by a neuropathologist for various pathological proteins, including p-tau. At the same time, our clinical team collects the donor's repetitive head injury, health, and neurobehavioral history by sending their families electronic surveys and conducting semi-structured interviews over the phone. Both the clinical and pathology teams are blinded to each other's findings. This is also when we administer a number of modified standardized scales to the informants of donors. These scales assess probable cognitive and functional impairment by using the cognitive difficulty scale, the functional activities questionnaire, and the brief metacognition index. We also assess neurobehavioral dysregulation, excuse me. Sorry, excuse me. By using the brief behavioral regulation index, the Baird impulsiveness scale, and the Brown-Goodwin aggression scale. We also assess some symptoms like depression and apathy by using the geriatric depression scale and apathy evaluation scale. And then we analyze brain regions involved in CTE with known roles in modulating cognitive, mood, and behavioral functions. Neuropathologists rated the density of p-tau in cortical and subcortical regions by using semi-quantitative rating scale ratings that range from 0 or none, or 3 or severe, and scores across all 10 regions were summed to form a global composite of p-tau severity, which gave us the overall p-tau severity in the brain. We also looked at regional p-tau severity, and here the dorsolateral frontal cortex and inferior frontal cortex were grouped to form a frontal cortex composite, and the CA1, 2, and 4 regions form the hippocampal composite. The rest of the regions were examined separately due to their more distinct neuroanatomical locations. And then experts assign each item of the cognitive difficulty scale to one of the 4 cognitive domains, specifically attention, memory, language, and motor. We then used multivariable OLS regression to analyze the data, where the independent variables included the global and regional measures of p-tau severity, with each p-tau measure included in a separate model. And the dependent variables included the clinical scales completed by informants, with each scale or factor score included in a separate model. The data that we got was very highly correlated, and to account for these correlations, we used ridge regressions to investigate which regions may be driving the associations, independent of the effects from the other regions. The sample was predominantly former American football players who played at the college level or higher, and there was other representation from sports like hockey, soccer, or wrestling. 15% of the sample also consisted of Black men. The mean age of the overall sample was 62, and here 63.2% of the sample had CT only, and the remaining sample had comorbid neurodegenerative disease diagnoses, with the most common being Alzheimer's disease. And additionally, all 4 stages of Alzheimer's disease were seen in this sample. We saw that cognitive impairment was, impairment was common in cognitive function, with 75.8% of the sample had elevated T-scores on the brief A MI, and 58.6% had an FAQ score of greater than 9, which is considered meaningful functional impairment. Symptoms of neurobehavioral dysregulation were present in 73.1% of the sample, indicated by a T-score greater than 65 on the brief A BRI. And there were high rates of elevated scores for apathy, as 80.1% had an AES score of greater than 34, and depression, where 78.6% of the sample had a GDS score of greater than 4. And then what we found was that greater severity of global p-tau was associated with worse cognitive and daily function. We can see that because the global higher rates of global p-tau was correlated with higher scores on the cognitive difficulty scale for the total score, and the other factor scores for attention, memory, language, and also motor. It was also associated with higher scores on the brief A metacognitive index, and higher scores on the FAQ as well. But greater p-tau severity was not associated with any of the neuropsychiatric scales. We used forest plots to show estimated effects of regional p-tau pathology on standardized cognitive scales. Here, the whiskers represent the 95% confidence intervals, and once again, it was a scale of 0 to 3. We found that p-tau severity in the frontal cortex, the superior temporal cortex, and the inferior parietal cortex was significantly associated with all the cognitive and daily function skills. We also found the same for the amygdala, where the p-tau severity in this region was associated with higher scores on all the cognitive and functional scales. But there was no association between p-tau in the hippocampus, entorhinal cortex, and locus ceruleus, and the cognitive scales. We did the same for the neurobehavioral scales and found that the p-tau severity in the frontal cortex was associated with the higher scores on the brief A BRI. But excuse me, but we did not find that regional p-tau severity in any of the other regions were associated with any of the neurobehavioral scales. And this is quite a busy slide, but what we can see here basically is that p-tau pathology in the frontal cortex showed significant associations across all scales of all the cognitive scales, and the inferior parietal cortex again was significant according to the ridge regressions for the cognitive difficulty scales and the rest of the factors. And it was the amygdala that was driving the associations for the functional activities questionnaire. We did the same for the neurobehavioral scales and found that the frontal cortex was driving them. We also did some sensitivity analyses and found that after accounting for comorbid neurodegenerative diseases, we continue to find significant associations, but with reduced effect sizes. There were some limitations as we couldn't administer these scales to the donors themselves, so there might be some measurement error and recall bias. And since this was a brain bank, there might be some ascertainment bias, but we've previously shown that selection bias does not negate associations in the Unite Brain Bank, at least with respect to repetitive head impact and CTE relationships. The current sample also consists of predominantly men who played football, and inferences to women and populations can't be made. And in conclusion, global and regional p-tau, especially driven by the frontal cortex, were associated with informed and reported cognitive and functional symptoms in an autopsy sample of brain donors with CTE. The regional p-tau aggregates, particularly in the frontal cortex, were an important determinant of symptoms of neurobehavioral dysregulation. And while there was support for neurobehavioral dysregulation, the effects were weaker and inconsistent across the scales. And this study does support the continued inclusion of cognitive impairment as a core feature of the TES criteria. I just wanted to thank my supervisors, Drs. Alasko and Mez, for helping me with this project, and everyone that works at the Alzheimer's Disease and CTE Centers for their support and work on the project, as well as those at the Concussion Legacy Foundations, and especially to the families that help with our research and contribute by donating. Thank you.
Okay, this brings us then to our final speaker and one of the last speakers of this conference, Martin Dahl from the Max Planck Institute for Human Development in Berlin, Germany. And he will refer about how longitudinal declines in locus coeruleus integrity are related to worse episodic memory.
Hello, thanks for having me. I would like to talk today about two neuromodulatory systems and how the changes in these systems are associated with how our memory changes in aging. I have nothing to disclose. Alzheimer's disease is characterized by the topographical spread of tau pathology from medial temporal lobe throughout the brain, and the spread of tau pathology is associated with both cognitive as well as neural decline. However, crucially, postmortem investigations dissecting the brains of people who died at different ages found that the first indications of this Alzheimer's-related tau accumulation can be found already in people who died at relatively young ages, and specifically in small subcortical nuclei deep down in the midbrain and brainstem. Based on this, in our research, we focus on two of these neuromodulatory systems. First, the noradrenergic system originating from the locus coeruleus, which you see here, which considerably declines with the progression of Alzheimer's disease. Here you again see the locus coeruleus, the core of the noradrenergic system, and here you see a second neuromodulatory system, the dopaminergic system, originating from the midbrain substantia nigra ventral tegmental area. These two neuromodulatory systems have in common that they produce certain neurochemicals, so-called neuromodulators, in very small circumscribed nuclei, but release them through the wide-ranging axons throughout most parts of the brain, and specifically in the medial temporal lobe and hippocampus, where both noradrenaline as well as dopamine facilitate synaptic plasticity and thereby long-term potentiation and memory. Crucially, through these wide-ranging axons, also tau accumulation can spread from the subcortical nuclei in the brainstem and in the midbrain to memory-relevant areas in the medial temporal lobe and contribute to memory decline. Therefore, these neuromodulatory systems are associated with the development of Alzheimer's disease, and we would like to to investigate their role in living people. There is a problem, however, which is that these neuromodulatory centers are just very small, which you see, for instance, here on an axial slice of a human brainstem. You can spot the fourth ventricle here as a landmark, and bordering the ventricle to the left and right, you see that colored area of the locus coeruleus, which has a diameter of roughly 2 millimeters. Similarly, here on the right-hand side, you see an axial slice of the midbrain, and you can spot this dark area corresponding to dopaminergic neurons of the substantia nigra ventral tegmental area. These two neuromodulatory systems were basically invisible for in vivo research for a long time. However, fortunately, about 15 years ago, the first imaging sequences were developed that now allow us to reveal these tiny subcortical structures as clusters of hyperintense voxels. So down here in the scan, you again spot the fourth ventricle, and bordering the ventricle to the left and right, you see these bright neurons or bright voxels corresponding to the locus coeruleus, and similarly here on the right-hand side, this brightness corresponding to the dopaminergic neurons. Crucially, post-mortem validations demonstrated that these brightness variations in the structural images correspond to the cell density or integrity in these neuromodulatory systems. Based on this, we added comparable sequences to the Berlin aging study, a longitudinal study of healthy aging in which we now have imaging data available for 2 measurement waves and cognitive data available for 3 waves, in a sample of roughly 300 younger and older participants. In the beginning, I would like to talk about cross-sectional findings, in which we linked in vivo proxies for these 2 neuromodulatory systems to aging cognition. And only later on, I will also talk about longitudinal findings, that is, within-person changes in these neuromodulatory systems. Coming to our imaging data. Here you see a standard T1 weighted scan zoomed in on an axial slice of the brainstem. You can again take the fourth ventricle as a landmark, this darkish area here, and bordering the ventricle to the left and right, you see this red dot, which is where we would expect the locus coeruleus to show up in dedicated imaging sequences. In this study, we added 3 such sequences, which are sensitive for the locus coeruleus. So here we have a T1 weighted fast spin echo sequence, and we can spot a clear hyperintensity, and we see a similar hyperintensity also in a magnetization transfer scan and in a proton density weighted scan. And again, we can take this hyperintensity as a non-invasive proxy for the cell density or integrity in these regions. Now, when we go from the brainstem a bit higher to the midbrain, here we find the dopaminergic neurons of the substantia nigra ventral tegmental area, and in red you see where we would expect that hyperintensity to show up, and here you see our 2 imaging sequences which pick up that hyperintensity. So in short, we use a multimodal approach to investigate 2 neuromodulatory systems in a large sample of younger and older participants. We developed a semi-automatic procedure to read out these intensity values for all of the different imaging sequences for all of our participants. Now, in this multimodal approach, our first question was whether using these different methods, we actually measure the same underlying construct. To demonstrate this, here you see our 3 imaging sequences for the locus coeruleus. On the x-axis, you see the standardized intensity, so our proxy for the integrity. And by comparing these histograms, you see this differs a bit across the different imaging approaches. However, crucially, when we correlate these values across different imaging sequences, we see that they are tightly coupled, indicating a high agreement across different imaging approaches. That means that using different imaging sequences, we actually measure the same underlying construct. Now, this is crucial because this now allows us to aggregate across different imaging approaches using structural equation modeling and estimate latent factors of locus coeruleus integrity and substantia nigra ventral tegmental area integrity that capture the shared variance across the different imaging approaches. And these latent factors are beneficial because they reduce measurement error and are more likely to detect true brain behavior correlations. To assess behavior in this study, our participants completed a very comprehensive cognitive screening, including 10 different cognitive tests, based on which we estimated latent factors of working memory performance, episodic memory, as well as fluid intelligence. In this study, we have a reference population of younger adults, which you see here, color-coded in gray. And in the blue tones, you see the older adults. And by comparing the distributions, you see that in each of these different cognitive domains, we see markedly impaired performance in aging. However, there is also a large spread in the older adult cohort, so large inter-individual differences, which we now would like to explain using our 2 imaging markers for these neuromodulatory systems. For this, first, we add our locus coeruleus factor to the cognitive model, and we see that those participants with a higher integrity in the locus coeruleus are those who are better able at solving the different episodic memory tasks, whereas those participants with a higher integrity in the dopaminergic brain regions are better able at solving the working memory tasks. Now, this may indicate that these 2 neuromodulatory systems play differential roles in aging cognition. To more directly test this, we now added the 2 neuromodulatory systems to the same model and searched for unique effects. And we additionally added a control region, which is the volume in the medial temporal lobe. So when we now have a look at associations between the locus coeruleus and episodic memory performance, we additionally control for the dopaminergic system, as well as the medial temporal lobe, as well as age, sex, and education. And we again see that the locus coeruleus is associated with episodic memory performance, and the dopaminergic system is associated with working memory performance, indicating unique effects of these 2 neuromodulatory systems. Now, the medial temporal lobe was initially associated with episodic memory performance when we analyzed it on its own. However, once we controlled for these 2 neuromodulatory systems, that association became non-significant. So in short, within one time point, in a large cohort of younger and older participants, we see that 2 neuromodulatory systems are differentially associated with aging cognition. Now, in a last step, we had a look at within-person changes in neuromodulation and tried to use these to predict the future cognitive status. For this, we computed latent change score models, which give us an individual estimate for the change in neuromodulation. And we see that the older our participants are, the more negative the change in neuromodulatory systems is, suggesting a degeneration of these tiny subcortical structures with increasing age. Next, we tried to explore the behavioral implications of this by linking within-person changes in neuromodulation to future cognitive performance. And indeed, we found that locus coeruleus decline was associated with future memory performance over and above chronological age. So to summarize, here we take a multimodal approach to investigate the noradrenergic locus coeruleus as well as the dopaminergic substantia nigra ventral tegmental area. And we find that these 2 neuromodulatory systems are differentially associated with aging cognition, with the dopaminergic system being more associated with working memory performance and the noradrenergic system being more associated with current as well as future episodic memory performance. With this, I would like to thank everyone involved in these studies and you for your attention, and I'm happy to take questions later on.
Thank you very much, Martin, for the excellent presentation. We are now moving to the question-answer session. Does anyone in the audience have questions for any of the speakers?
Yeah.
Okay, we can take question from microphone 2 first.
Okay, thank you. It was a great session with a good variety of many talks. I'm Suzanne from the Alzheimer's Center Amsterdam, and I have a question for you, Farah. So you showed that I think p-tau pathology in those CTE cases was strongly associated with cognition, but only moderately to neurobehavioral dysregulation, even though I think the percentage of these symptoms were pretty high. And I was wondering if you can speculate on if it's maybe due to other pathologies or maybe non-biological environmental risks.
Yeah, so that's a really good question. That's what we're trying to sort of figure out right now. It could be various factors, like yes, like for example, like aggression is really well known and commonly associated with CTE, but it's hard to tell if like the people that play sports are already naturally aggressive. So there are some confounding factors there. And then we did like a limited number of tests, and again, these tests weren't administered to the donors, so there might be things that we're not capturing that might be associated.
Thanks.
Mm-hmm.
Hi, my name is Toon Rense. I'm also from the Alzheimer's Center in Amsterdam. I have a question for the last speaker. Very interesting talk, thank you. Also, after having heard all the talks yesterday about locus coeruleus, this is a very new field for me, so I was wondering, as a neuropsychologist, did you also look at episodic memory on a sort of subtest level, since episodic memory consists out of consolidation, retrieval, and recognition of information, and what can be attributed to the locus coeruleus itself, and what maybe is located in other parts of the brain?
Yeah, good question.
Now.
Okay, I think this works now. Yeah, I think your question is totally valid. In our study, we rather took the approach that we tried to aggregate across different tasks to get a bit more power and to be able to generalize more. In previous studies, we saw that especially verbal learning and memory, so for instance, the Ray test would be associated with the locus coeruleus. I think from animal research, we know that these neuromodulators play a large role on consolidation of hippocampal memories. But I think there's always a compromise, whether you try to generalize more or whether you try to explore the specifics. And I think in a future step, we should go that route. Thank you.
Yes, microphone number 4.
That's this one. Daniel Ferreira from Karolinska Institute. I really enjoyed this session, very inspiring. Thanks to all the speakers. I have a question for Michel Grotte. Well, first I wanted to celebrate all the efforts you are doing to expand the use of FDG to interrogate TDP-43. I think that's important, and not many labs are doing that, so congrats. So I think your findings could have some clinical application in the future, perhaps, and a common clinical differential diagnosis in those patients is hippocampal sclerosis. I was thinking if you see any opportunities for magnetic resonance imaging, perhaps to assess hippocampal sclerosis and maybe stratify people with TDP-43, FDG pattern, hippocampal sclerosis in MRI, since there is a high association between the two.
Wow. Hello? Now? Yeah, okay. Yeah, that's a very good question. So I have to say this, the limbic TDP-43 pathology late is really highly associated with hippocampal atrophy on MRI. So whether they are pathologically diagnosed also with hippocampal sclerosis or not, virtually all of these autopsy cases with relatively pure late have severe hippocampal atrophy. And that's, so when we tried to use some, just focused on the autopsy cohort to try to differentiate between these relatively pure late cases and the relatively pure AD cases, adding MRI or specifically hippocampal volume Two, our FDG PET measures didn't really increase the discriminatory ability of the models but to discriminate between these two pathologically defined groups simply because they all have quite elevated degrees of hippocampal atrophy. But yeah, so I think in a clinical setting, since you usually get an MRI scan and you have an amnestic syndrome, patient presenting with an amnestic symptom, you're not sure what it is, you usually get an MRI scan, you see there's hippocampal atrophy, and it's from that point when you want to know whether it's AD or late, basically.
It's microphone number 6.
Hi. Thanks for a great set of talks. Question for Martin. I think, if I remember correctly, the cognitive domains that you were correlating in your study were episodic memory and executive function. Wondering if you saw similar results with any tasks loading, particularly on attention, given the neurogenergic effects of ELC and the association with attention.
Yeah, good question. I mean, from animal research, we of course know that the noradrenergic system plays a crucial role in selective attention. And we often also see this role when we take more time-sensitive measures of the noradrenergic system, like, for instance, pupil dilation, the P3, event-related potential, or event-related desynchronization. In this study, we had 3 cognitive domains. We had episodic memory performance, working memory, and fluid intelligence. And I think the working memory factor especially would be comparable to attentional performance. For this factor, we see that it is more related to the dopaminergic system, potentially due to dopaminergic neuromodulation facilitating prefrontal delay activity, as shown by Amy Arnston and others. It's a bit challenging to say why we don't see that link for the noradrenergic system here. It could be that the noradrenergic system is more involved in reorienting attention, like, for instance, in an oddball task where you would need to switch between different attentional sets. And here we had tasks more like a number n-back task, so a bit more keeping information active in mind and modulating it.
Thanks. Yeah. Mic number 6.
Hi, Matej Peronik, University Medical Center Ljubljana, Slovenia. I have a question for Dr. Grote. Since we have seen also in the talk today, there is a great overlap between AD and late pathology, so they don't they co-occur often. So I was wondering if you were if you could include an intermediate category in your classification algorithm, so not just have AD-like and lady-like, but some that are having both.
Yeah. That's a very good question. I actually, like understanding this classification procedure, it's really, we're trying to find a match between, so whether an individual profile is more related to one pattern than to the other, but there are a whole lot that are just in between the two. And so what we did, we were just, basically we set a cut point in the midline just between the two, but so both sides pretty much include like borderline cases where you have these two. We did analyze these, let's say, mixed cases, like around the median cut point, to see whether these could reflect comorbid cases, and they did show some like intermediate characteristics. However, we're not really sure if this approach could work this way, because, for example, these pathology studies have shown that it's nonlinear. Or that they're synergistic in a sense that mixed AD plus late cases usually show the steepest curves of cognitive decline, steeper than in late, or even steeper than in AD alone, and much steeper than in late alone. And we didn't see that for this mixed pathology group. But then again, it's also tough in these classifications to control for the overall degree of atrophy or hypermetabolism in this case. I think it's a really a big challenge, a much more complicated problem, let's say, to identify these mixed pathology cases. And this is really where I think eventually we will need some molecular biomarker for TDP-43, because in these mixed pathologies we get to limitations of these indirect topography-based neuroimaging approaches.
I think we can have one last question.
Hi, my name is Heidi Jacobs from MGH. I have a question for Martin. There's some beautiful work from Deborah Bengasser showing there's a differential involvement of locus coeruleus neurons in males versus females. And I was wondering if you saw some sex differences in the relationship between locus coeruleus and cognition, or in the substantia nigra versus cognition.
Yeah, very good question. We do definitely see sex differences in the sample. We had a previous study also in a similar cohort, just cross-sectionally, that was led by Shelby Bachman from USC. So she explored the association between RC contrast and cortical thickness, and she found that this differed quite a bit, that there's association between males and females. In this current study, we see that especially verbal memory differs between males and females, as has been reported previously, but we also see sex differences in the locus coeruleus. In general, the effects hold when we control for sex, so I think it's not purely driven by this, but it's definitely part of the pattern. And I mean, as you mentioned, there's also some animal research indicating this.
Thank you.
Okay, so I think in the interest of time, we conclude this session. Thanks very much again to all the speakers. Thank you for attending this last session, and hope to see you all next year.